welcome you all to our March Discovery Lab, the topic being oysters. And so again, we are educators at the Chesapeake Bay National Estuarine Research Reserve in Virginia, housed at the Virginia Institute of Marine Science. And if you've joined us for our January and February lab, you know we've been doing them virtual over Zoom webinars rather than in person. And we've had a lot of great feedback about these programs, and we're really excited for this evening with you all. Oh, and let me progress to the next slide for you all to see. So this evening we have Dr. Ryan Carnegie with us. So he's going to give a presentation about oysters and some of the research and some of the work that he does here at the Virginia Institute of Marine Science. After that, you guys are able to ask some questions for him. And we ask if you do have any questions to use the Q&A box that's located either at the top or the bottom of your screen. And that way we're able to see when those come in. If it's something that Sarah and I can answer, we can do that as well. And if you just have any questions, please leave them there. And you can also use that chat as well. Then we'll be doing a live oyster reef habitat exploration here in our lab at CBNERR, Virginia. And we'll be using a microscope exploring some of the cool animals that live in our oyster cages. And then we're going to wrap up this evening with some oyster trivia. So if you guys liked the Kahoot trivia before, we got another round for you. And then we will be emailing out those additional resources that Maggie and Sophia created, those awesome resources that you guys are going to be able to use after this program that you guys can do from the safety of your home with your families. And so with that, I also want to let you guys know that we have one more Discovery Lab coming up next month. So that'll be on April 20th, again at 6 o'clock on harmful algae blooms. And that speaker is going to be Marta Sanderson. We'll be doing demos and trivia and activities for you all with our eco ambassadors. They'll be kind of taking the helm there. And we'll be sending out information about that registration. You'll get a link just like you signed up for this one, very, very similar. And for those updates, if you want to follow us on our social media, we have our Facebook, our Instagram, and Twitter. And so those are ways that we can keep you guys up to date on any programming, and those registration links are usually posted there, along with the VIMS social media account as well. And with that, I do want to introduce our guest speaker for this evening, Dr. Ryan Carnegie. So he is a professor, research professor here at the Virginia Institute of Marine Science in the Shellfish Pathology Lab. And so he's going to give you guys a rundown about some of the basics of oyster ecology and some of the work that he's done. And with that, I'll go ahead and turn it over to him.
Okay. Thank you, Tara. So let's see, I'm sharing, continue. Okay, Tara, is that all set from your end?
Yeah, looks great.
Okay, perfect. All right, well, thanks everybody for tuning in tonight. I'm super happy to be here with you to talk about oysters and oyster diseases, which maybe in some twisted way, that's one of my favorite topics. But this is what I spend my career doing, is trying to better understand these parasites and pathogens that are affecting these very, very important marine species. So let's see, moving forward. Yeah, so I just included a few pictures here. I mean, oysters and charisma are not two words that you often hear in the same sentence, but these are charming and very prominent elements of our estuarine systems in many, many parts of the world. And I highlight just a few of them here. You see Australia and New Zealand are represented, but there are also two oysters that are right here from on our east coast that you might not know about. An import from Europe that lives in New England at the lower left of the European flat oyster, and crested oysters, which is a small non-commercial species, a beautiful little oyster living in North Carolina and further south. So these are, again, prominent elements of our estuarine animal fauna. So I'm going to structure my short presentation around three basic points. And the first of these is that oysters are important. They're providing essential ecological services, which is what you're going to be learning about in the Discovery Lab tonight. And these includes the structure and the habitat they provide to marine systems that, certainly in our part of the world would otherwise be mostly just soft mud. So they provide an important element of hard structure to these estuarine systems. They're important in ceston filtration, which means they're filtering and consuming and dragging out of the water column all of the microscopic plants and bits of non-living matter that's drifting by in the currents. They're grabbing that and directing it to the benthos. Benthic pelagic coupling in general, providing connections of many, many different types in terms of energy flow and nutrient flow between the bottom and the water column in our estuarine systems. And we hear a lot about ocean acidification nowadays, and these oysters producing these very large amounts of calcium carbonate do provide an important element of carbonate buffering in our systems as well. Of course, they're important because they're a resource for coastal communities. Oysters have been consumed by coastal communities for millennia, really forever. As long as people have lived on the shores of temperate estuaries, oysters have been consumed by these people. And certainly today in our region, oyster industries are vital to the well-being of coastal communities. And we know what the effects of diseases have been on these communities as we've seen them fall into despair in many cases by the collapse of oyster fisheries in the last century. So we know they're important for this reason as well. And we know furthermore that oysters and other bivalves like clams and mussels are only going to be more important for our food future. Bivalve mollusks like oysters and clams, they really need to be an increasing part of our collective societal diet. They're locally produced, so we could ship them all over the place, but generally these are species that are growing on the shores, and where we acquire them, there are not distribution chains that span across continents bringing oysters to our markets and our tables. They're very nutritious and they're ecologically sustainable in ways that many of our food industries are not. And this is just a graph here that I found just today from a 2020 dissertation at the University of Washington. And I highlighted with an arrow where bivalve mollusks fall on this graph, all the way at the left, producing relatively low amounts of greenhouse gas emissions per nutritional score. So they're very nutritious, and relative to their nutrition, they're producing very, very low levels of greenhouse gases. So this is speaking to their ecological sustainability. So the second point I would raise is that oysters have been troubled for much of the last several hundred years of their history. These are a couple of images showing what oyster industries were up to maybe 60 or 70 years ago. So oyster populations in places like the Chesapeake Bay and Delaware Bay have been significantly destroyed since the 1850s through overharvesting, pulling way too many oysters out of these populations off the bottom of these estuaries. And with the removal of oysters is the removal of habitat. So we're losing the animals and we're losing the structure, the physical environment that they need for the next generations to settle on and so on. So this has been really tremendously destructive and disruptive to their populations. And again, this started around the 1850s, really in a serious way, when we had both railroads to carry away from our shores more oysters than we could consume locally, and refrigerated rail cars to transport them safely to the middle of the continent. And this was going on in Europe as well at the same time. But in the 1950s, this is really where locally we had the emergence of infectious diseases affecting oysters in our areas. The first of these on the left is Haplosburium nelsoni, or MSX, and I've highlighted with the arrows a couple of different cell forms of this parasite. And on the right, Perkinsus marinus, or dermo, and I've highlighted a couple of cell forms of Perkinsus over here on the right. And these are both protozoan parasites, single-celled organisms that are living inside the oyster tissues and consuming them from within, and often causing very high levels of mortality in the oyster populations along the way. Neither of these parasites is a human health concern, I need to say. I often forget to mention that. These are not human pathogens in any way. They just kill the oysters. So just to say a little bit more about these, MSX, this is an exotic pathogen that emerged in our system in 1959, in Delaware Bay in 1957. This is an invasive pathogen from Asia. It kills very quickly, developing infections, causing mortality within just weeks to months, killing oysters of all sizes and ages. And in the early years, when it first emerged, it killed more than 85% of oysters in our populations very, very quickly. MSX transmits through some intermediate host or hosts. We have no idea what they are. Some small worm or copepod or something that's living in oyster reefs and oyster culture systems. And dermo is the other major disease. This is a native pathogen, probably has always been here. Historically, this was more of a chronic disease, so not causing rapid acute mortality on a scale of weeks to months, but killing more after 2 or 3 or 4 years of exposure. The initial mortality caused by dermo was on the order of 30%, typically in the late summer and fall. Dermo is directly transmissible from oyster to oyster, so it infects one oyster from the neighboring oyster on a reef. And this was a relatively minor disease until it intensified in the 1980s, suddenly causing very, very high levels of disease and mortality. So this is just a bit of an introduction now to sort of what we're doing and how we're looking at these systems. And this is how we sort of view them in my research program. We try to understand how the oyster interactions with these pathogens are changing over time, evolving over time. This is the evolutionary ecology of these relationships. And the answers to these questions and how all of this is changing over time are a key, actually, to understanding our own history with this important marine system. You know, we shed light on the events of the last 20 or 30 years by virtue of what we're learning in terms of the evolutionary interactions of the animals and their pathogens. And having better knowledge on this points the way forward to effective resource management for the future. And I'll just say a little bit more about this. This graph is it's produced from data from the Virginia Marine Resources Commission. And these are the landings in Virginia of oysters in bushels per year since 1958, going into the early 2000s. So in the red is off of private leases. This is early aquaculture, extensive, mostly planting on the bottom of the bay. And in the blue is public reefs. These are wild harvests of natural oyster reefs. And we can see the impacts of MSX mostly on the private aquaculture beginning in 1959, and then the intensification of dermo in the 1980s. So because of where we were from the late 80s into the 90s and the early part of the 2000s, many of you probably remember that we were contemplating introducing a non-native oyster, which is in this picture here, Chrysostia eriacensis. And this made really perfect sense at the time because, the oysters have been living with these two parasites for decades or more at that point. And if anything, I mean, you can look at where the graph is in 1998, for example. If anything, they were doing worse after all that time than they had been before. So it was crazy to think they were going to get any better. So why not introduce a non-native? And we came very close to doing that. But what we've seen in my laboratory's research is that this great intensification of dermo in the 1980s was not just due to climate factors. It was warm, it was dry. It was due to the emergence of an entirely new strain of the parasite that showed up in the 1980s or emerged locally in the 1980s and completely took over, looking different, dividing in a different tissue within the oyster bodies, causing much higher levels of mortality. This is something that we didn't know until recently, but it helps us sort of interpret all of this earlier history in a totally different way. Of course, the oysters were doing terribly in the early 1990s. And it wasn't because they were hopelessly stuck in this long-term relationship that wasn't working for them with the parasites. They were being challenged by a brand new pathogen, effectively, beginning in the late 1980s, and they just hadn't adapted to that yet. So that's just one example and one very prominent example in our research of how we're using these evolutionary perspectives to learn more about sort of the history, the historical ecology of our systems. The final point I'll make is that the oysters are coming back. which is important. And I would certainly like to finish on a note of optimism as I wrap up this short presentation. So this graph you've seen already a couple times. This is the last time I'm going to show it to you. But I've made available the right end of the graph at this point, so you can see where we are in more recent years. We see that we are basically witnessing an increase in oyster abundance in our system. Now, sure, part of that is that we're growing more oysters in aquaculture. And many of you, if you're thinking about oysters, you know you're seeing more oyster aquaculture, you're seeing more oysters served at raw bars and in the restaurants and so on. But the increase is happening in the blue too. This is oysters in wild reefs. This isn't just people growing oysters. This is just the oysters in the natural systems themselves. So what's going on here? And let me just open all these up at one time. Is this something that we're just doing in terms of oyster restoration in one place that's working? Well, these are data in these graphs here from my colleague Roger Mann and Missy Southworth from our molluscan ecology monitoring program, looking at oyster abundance across 5 different Virginia systems over time, since the 1980s to about 2018. We could see that this increase in oyster abundance, which is what you're looking at here, these are oyster numbers per bushel, is increasing not just in one place by virtue of some of the restoration activities we're doing, it's increasing everywhere. And if that's happening, it must be something that's more inherent to the animals themselves that's improving, and not just something that we're doing. We're doing restoration more effectively, for example. And this is another area where we've applied sort of this deep dive into the biology and the ecology and the evolutionary biology of these animals to shed light on this. And just, very briefly, this is my graduate student who just finished Lauren Huey's work, where she found that in 2003, oysters started developing a quantitative improvement in their ability to reproduce in the face of these oyster disease challenges. And what we're looking at on the lower right, these are just oyster eggs, within their bodies that they're ready to spawn and release, that they were not able to produce effectively at one point because of the diseases, but that all of a sudden they were able to start doing a better job of that, contributing to this general improvement in the oyster resource in our system. So basically, this is my conclusion. What are we learning, about a way forward from this quick snapshot into the work that we're doing in this laboratory? First, long-term evolutionary perspective is actually very important in highlighting how dynamic this system is. And it also highlights the resilience of the oyster, that this animal that we once wrote off as a lost cause is actually taking quite good care of itself in this system. And we just need to figure out a way to help it do that. It speaks to the importance of conservation underlying all of the management we're doing. For example, sanctuaries and so on, to allow the animals to work things out evolutionarily with these pathogens without our intrusion in the process, removing animals from the system that might have had a chance to spawn another 8 times and pass on the good genes, to continue this development. It also highlights, in the broad scale of the improvement that we're seeing in the oyster resource, it highlights the potential value, not just of sort of massive investments in restoration in one place, but the potential value of dispersed community level activities that we might be doing on the shores of all of our, waterfront communities to try to help oysters, recover. That all of these activities that we're doing, even at very small scale, might be much more important and impactful than we think. So with that's all I have. I will be happy to take a couple of questions, which I see I did manage to leave some time for. And I threw this figure up here. It's always good to maybe prime things a little bit with some questions. So, some possibilities, what do oysters like to settle on? What's the deal with pearls? Do oysters produce pearls? Why is Ryan eating oysters in the middle of the summertime when we're only supposed to eat oysters in months with R? Where's the head and the brain in an oyster? Do they have those? Anyway, I'll leave it at that. Thank you.
Well, thank you so much. And we do have a few questions that people are very eager to ask. So someone's asking, where do oyster pathogens come from or originate?
Well, so the one pathogen came from Asia. So MSX is an Asian import. It probably either came over with Pacific oysters from Japan or Korea. So this is a species that was experimented with in the Chesapeake Bay region and to the north and to the south, pretty much everywhere at one point on the east and Gulf coasts. Or it came over with ships returning from the Pacific theater after World War II or the Korean War. We will never be able to resolve the answer to that. The other parasite, dermo, has probably always been here.
So someone's also asking, where would you find the healthiest oysters in the entire world? If you had to take a guess.
Well, that's a good question. You know, oysters have problems in a lot of places. You know, and one of the things we think about is, you know, why do oysters have so many more diseases around the world than a lot of our other marine bivalves, for example? And part of that has been that I think they're so cooperative about closing themselves up very tightly and allowing us to move them from place to place, you know, in a way that a lot of stuff that we can't do. You know, scallops will gape and die, you know, and they're not going to survive a long plane trip or, you know, centuries ago, like a long, you know, ride in the bottom of a ship or something. Because oysters close so tight, we can move them from one place to another, moving diseases around, creating new encounters between the animals and pathogens that might occur in that new population that they've never been exposed to, creating these emergences of disease all over the place and you know, at different times. But there are perfectly healthy oyster populations all over the place. Here in the Chesapeake Bay region, you know, certainly, you know, all around the world. Atlantic Canada, you know, doesn't have, you know, I think by virtue of their cooler environments, a lot of the cooler areas don't have as many of the disease problems as the warmer areas, if I had to generalize it all.
Perfect. This is a pretty interesting question. So you've talked about how oysters are filter feeders, but has there been any evidence that oysters have the ability to filter out things like microplastics out of the water?
Well, there's been work done on that, and depending on the size of the plastics, these marine bivalves they're definitely interacting with plastics on some level. I mean, certainly, you know, stuff that is, you know, half of a millimeter or something. I don't have the sizes of my marine plankton committed to memory very well, but certainly bigger plastic bits, you know, are going to be big enough to be captured on their gills along with the food that they're feeding and filtering. Smaller stuff might get passively ingested. There's a potential that all of it could be ingested and passed through. There's work being done on the assimilation into tissues. You know, some of the microplastics and nanoplastics can get into the bodies, and does that cause any health problems and so on. So there's attention being given to that. I should say that there's no evidence that we have yet that this poses any acute health issues for the oysters themselves, nor does it pose any issues for human consumers. You know, we unfortunately have a lot of exposure to plastics and microplastics just in our day-to-day lives, you know, hanging around, lying on the carpet at home, for example. You know, and so I think it's the sources of microplastic that we're encountering are way beyond what we would be experiencing from consuming oysters.
Yeah, exactly. And then also, you've talked a lot about the different diseases that affect these oyster populations. Are you also seeing that these diseases are found in oyster farm growths or oyster farming?
Yeah, so you know, all of these diseases, they can be found in both farmed and wild environments. We are looking carefully at you know, what's happening in farms versus the wild. We published an interesting paper a couple of years ago. You know, one of the things that a lot of people think is that you know, well, you know, a farm, you're growing oysters at high density. Dermo in particular, like I said, it's directly transmitted from one oyster to the next. So, you know, like packing a bunch of kids in a kindergarten classroom, you know, all those oysters in a bag is just going to create more disease transmission. So obviously farmed oysters on farms are going to be sicker from dermo disease than, you know, out on wild reefs. Well, the thing is, you know, dermo takes a little bit of time to attack these oysters. You know, it takes a couple of years of exposure before it gets to a high level. And, you know, the oysters on farms, you know, when they're grown intensively in bags and cages, they're typically marketed after like a year and a half. So they're out of the water before disease levels on the farm peak. It's also the case that, no oyster farmer, is going to be making money if they're letting all their animals die of disease before they can get them to somebody's table, or the market. So it turns out, and this was the point of our paper, that because of the nature of the way the diseases sort of attack the farms, and it takes a little bit of time, and the fast rate at which the growers are planting and then removing stuff, farms are actually a sink for the parasites. They're filtering, and then they're removing them from the systems, possibly to the benefit of the wild populations. It's cool stuff.
Yeah. And we have time for a couple other questions. And if we don't get to everyone's questions tonight, because you guys are asking a lot, which is great, we'll be leaving his email in the chat for you guys. So if you didn't get your question answered, you're more than welcome to email Dr. Carnegie, and he will get in touch with you. But we'll have time for two more. Someone was asking about where exactly oysters grow. So can they grow in really cold places, say like the polar regions?
We have some oysters that tolerate colder areas, but they tend to sort of peter out. They don't occur that far north into Canadian waters in the Atlantic. The Pacific waters of Canada are a little bit warmer. There are oysters being grown in Alaska, for example. In Scandinavia, Norway has some oysters, but not too far north. Iceland is thinking about growing oysters, but it's you're starting to get into like slow growth rates, and when you've got a crop that's out there for a long time, there's a lot of things that can go wrong with it. So there's some stuff that needs to be worked out, but it's more like the temperate areas to sort of subtropical, where most of the oyster activity is.
Just right where we are. And then the final question is going to be about, again, going back to like farming for oysters and wild oysters, if there's really a difference in the quality of the oyster, especially when it goes to, say, the consumer.
Well, that's a good question. There are some fantastic wild oysters out there. The best quality wild oysters will tend to be on reefs that are not super dense. Like, for example, the James River, we have tremendous oyster populations in the James, but the populations are so dense that the oysters grow like fingers sticking up straight. They're so almost starved because they're competing with each other so intensely for food, and their condition isn't great. In parts of the Rappahannock and the Piankatank, for example, where the densities are lower, they have more space to grow and filter and develop better condition. And certainly in aquaculture, the growers are focusing on putting all their effort into developing a really exquisite product. So I would say on average, the better product is going to be coming out of aquaculture, like, for sure.
Mm-hmm. Yeah. Perfect. Well, thank you so much for coming to speak with everyone this evening. And we love that everyone had so many questions to ask. And again, we can put, Dr. Carnegie, if you want to put your email in the chat, feel free, and we can do that as well. So if you have any further questions or you're really eager to learn more about his research and the work that he does, feel free to shoot him an email. Sarah and I can also help you get in touch as well. But we definitely want to thank you for visiting with us this evening, and we really appreciate it. And we hope you guys have learned a lot from him. And so thank you. If everyone could give a virtual round of applause, that'd be great.
Thank you.
Awesome. And so with that transition, guys, so we're going to move into looking at an oyster cage habitat. And so what I'm going to be doing is I'm going to pull up a little PowerPoint, very short. And so the Chesapeake Bay National Estuarine Research Reserve in Virginia, we normally do a field trip program in person with students who come to our site, and we wade out into the York River, and we have a series of oyster cages. And we leave them out there for about 6 to 12 months. And these oyster cages will bring back into our lab, and the students will have the opportunity to really explore and identify some of the critters that are found in these habitats. So We've talked about oyster reefs, and we like to say that these oyster cages are just a miniaturized version of an oyster reef. It's its own habitat just within a mesh cage. And so this is Matthew. He's one of our educators. This picture was taken as he was removing the oyster cage from the York River and placing it into a bucket, which then we bring back into our lab. And so you'll notice here 2 images, and that oyster cage, again, these are important because they do act like an oyster reef, where oysters can have some hard surface to attach to. And these are really important because, one, they allow the oyster population to grow. It also allows the oysters to filter water, since they are filter feeders and we want nice, clear, beautiful water. And then it also provides habitat, not just for oysters, but for a lot of different macroinvertebrates and fish and plant life, all within this habitat. And so as we bring it in, we place it in a bucket of water, and that water is pulled from the same environment that the cage was. We put in a bubbler, and so that aerates the water, especially if we leave it overnight inside. We want the temperature of the water to be the same, and we want to make sure we're keeping the water moving for the animals inside. We then use scissors, and we cut these zip ties that are keeping the lid shut, because if it was open, they would go everywhere. So once we've uncut those zip ties, we can open the cage, and on the right-hand side, you can see we have a whole pile of oysters there. And this is great because this is where we actually get to go in and explore what exactly is inside this oyster cage and this oyster cage habitat. And that's what we're going to be looking at this evening. And there's a couple ways that we do this. You can use something like an ID sheet. So this is pictured on the left-hand side. It just shows an image and it gives you the name of the animal. So this is super easy. You just match the organism find with the image on the ID sheet. But we can also use, for more of our older students, a dichotomous key. So this is another tool to identify these macroinvertebrates and organisms that live in this oyster cage. They don't have pictures, so you have to rely on descriptions and follow the order of the descriptions to find the organism that you're looking for. And so we recently pulled out a habitat cage for you all to see today. And so I'm going to stop sharing my screen very quickly so you don't see a PowerPoint. And so we are going to be using a microscope that is attached to my Zoom computer. So I'm going to plug that in. And when we use this microscope, we're going to try our best to look at some of the smaller organisms, and I'll tell you a little bit about them. As I move some of the dishes around, it might make you feel a little dizzy just because it is zoomed in so much. So I'll try my best to keep that at a minimum. We don't want anyone getting dizzy. And then I'll show you some of our larger organisms that we found as well. So I'm going to switch over my camera so you guys are going to be able to see this. Awesome. So on my screen, you should be able to see not my face anymore. So you'll see some green material there. And this is actually as zoomed out on the microscope as I can possibly get. But this zoomed in is not going to be a type of animal, but rather a type of plant that we find pretty common in our oyster cages. So this is called sea lettuce. And it's called sea lettuce because it is almost the characteristics of lettuce itself. And it is a form of algae. And it's very, very abundant. And it can be produced in very large quantities. And sometimes if we have too much sea lettuce, what can happen is that can be an indicator of a lot of nutrient pollution and an unhealthy ecosystem. And I'll show you kind of what it looks like not under the microscope. So this is sea lettuce. So as you can see, it's stuck to the container because it's a little wet, but this is a very leafy, almost does look like lettuce. So that is not under the microscope. So you can definitely see how zoomed in it was on that microscope there. And so I'm going to switch back and we're going to look at our second organism. Back to our microscope. And I'll see if this one will show up. This one's going to be a little tricky because this one will move. I have to find it first. The tricky part. There it is, part of it. Awesome. So you guys should be able to see a little bit of that. It's a little stuck there. But this is a type of polychaete worm. And we call this a clam worm. And as you can see on the side, they have these little pink bristles almost. And so as they go along, these are really common in our oyster cages. They feed on a lot of dead material. They can feed on some plant life as well. Let's see if I can get near the head. Sorry if I'm making y'all dizzy. I'm trying my best. But you can see that nice clear red line right down the middle of the body, and they have these segmented sections. And again, those pink bristles along the side. And they do have a head and they have eyes, and they use their antennae to find food to bring it in. And they can vary in size, but again, they're pretty small. This one is actually one of the largest ones that I have seen, and I'll show you in just a moment without the camera. But they can be pretty cool right underneath there. So I'll switch back real quick. And I'll see if I can show you without the water. Pick them up a little bit for you guys. So this is a polychaete worm or clam worm. So as he drips it back down into the water in the container, you can see how long he is. And yeah, usually we find them to be a lot smaller than that one there. But again, they can be scavengers and they prey on a lot of different items within those oyster cages. And the clam worms tend to be more red in color than any other. And then we're going to show you, we'll see the skillet fish first. So we'll switch to our microscope one more time. Oh, and he popped out. No, come on, buddy. It's fun working with wildlife under a microscope. They like to jump out. So this one, you can kind of see there. You can see the skin there. So this is a skillet fish. And so you might have seen videos of them before or have learned about them or seen them. And I'm going to show them off camera in just a moment because it's a lot easier. But oh, there's a good image. You can kind of see one of the gills going and one of those fins as well. But you notice just the texture of their skin's very slimy. They have those speckles as well along their skin. But if we were to look at them normally, and I'll actually just hold him for this. He'll be okay for just a moment. And I'll tell you why they're so difficult to grab. There we go. Protecting my keyboard. So this is a skillet fish. They are shaped like a skillet, like a frying pan, very flat. And the reason he was so difficult for me to grab, and I'll place him in water for this, is because they actually have a suction cup on the bottom of their body. So he's able to stay very tight and stick to the side of a container like this one right now. Oh, he doesn't want to stick to the side for you guys. He wants to swim out. But they can stick to the bottom of boats and they can be really difficult to pull off because that suction cup on the bottom of their body is pretty strong. So if he goes back on the side, I'll show you guys in a minute. But I do want to show you a couple more. And we'll try these under the microscope as well. Move my trash bag. So we'll go back to our microscope. Perfect. So, well, he was for just a moment. So this is another type of fish that's fairly common. This is a naked goby. And again, fish can be hard to see under here because they are moving around and don't want to stay in one place for too long, but you can see an eye right there, 2 eyes right on the top of their head. These are going to be a little bit smaller than our skillet fish. And the reason we call them naked gobies is because they don't have scales. There are fish that exist that don't have scales, and rather they have a protective slime, making it difficult for predators to go after them. And we're not going to hold him just because he's in more of a shallow dish and he wouldn't stick to my hand like that skillet fish. And we have one more to see under the microscope. So let's give it a try. We'll see if we'll get it. We can find it. He's right on the edge there. Oh, we can kind of tilt him. It's difficult to see, but that's one of the eyes of this animal. Let's see if I can see a part to give it away. These animals are on the move and don't want to be under the microscope. So this is a mud crab. And you can kind of see some of the legs there. They can be a little trickier to see under the microscope. So I'll show you what they look like in person. Grabbed him by his apron. So this is, like I mentioned, a mud crab. So they are not our blue crabs that we tend to find, but mud crabs, they tend to be more brown in color, sometimes black, so they blend in really well with that oyster cage habitat. And they are scavengers too. They pick off algae, small fish, other crabs even, and they don't get too large, at least the ones that we see around here. And there's many different species of mud crab that exist, but this is about the relative size that we see, so not too big. And they still do have the pinchers and the claws like we see with our blue crab species, but the coloration and the size is definitely going to be a lot different than our blue crabs that we see. And we do find blue crabs in our oyster cage habitats, but that tends to be more during the warmer months of the year. And then the last one I want to talk about, we won't show under the microscope just because it's too big, but it's the whole topic today. It is our eastern oyster. So again, let me get my protective covering. So this is an eastern oyster. This is the oyster species that we see mainly on the Chesapeake Bay in our oyster cage habitats that we have on the York River. They are a bivalve, and so they do have a hinge right here and 2 different shells. And the shell size, the shell shape, let's try saying that 5 times fast. They tend to have one that's more shaped like a cup and then a flat side to the oyster. And there is an animal, the oyster is living inside of it. It uses a muscle called an adductor muscle, and that keeps the animal attached to the shell. It keeps the shells close together and nice and tight and sealed. And they still are filter feeders. And you'll see this in our trivia game where these animals are able to filter a lot of water throughout the day. So areas where we see a lot of these oysters and these oyster reefs, we see clear water. We see water with really good water quality parameters like pH, salinity, turbidity, and it creates a really healthy environment. And sometimes you can even see, it might be hard to see on this one, you can see barnacles grow on these oysters and other organisms as well, because they tend to be pretty stationary later in their life. When they are in their younger phases, they do drift until they find that hard substrate surface to attach to. So this is definitely the star of the show this evening and the main organism that's going to be living in our oyster cages. But from what we just showed you guys, you can tell that there are many different types of animals that live in these habitats. And I was even surprised because the water temperature has been pretty chilly. So we weren't expecting to see a lot. So it's great that we were able to see some fish species, see some of those worms. And there were a ton of different mud crabs in there as well from different sizes. So definitely a nice healthy environment. And Once we're done with the oyster cage, we attach more zip ties. We walk it back out to the York River and we place it where we left it. And most of the time we do want to maintain them. We want to clear them out and make sure they're not overcrowding. And there's lots of space for those organisms to thrive. So like I mentioned, normally this is a program we do in person and the kids get to really handle the oysters and the oyster cage and really dig through it to find some of the animals. But we hope this was kind of a different way to view them, especially with the microscope. It can be a little tricky with technology, but we definitely got to see some cool things. So I hope you guys enjoyed that. And if you guys have any questions about that activity or about how these oyster cages work, you guys can drop that in the Q&A or the chat and we can answer those. But I do want to go ahead and kind of round out this evening with our classic Kahoot trivia game. And so as I pull this up, let me go ahead and get it all ready for you guys. And for those of you who haven't played with us before, I'll go through those rules again with you all. But let me share my screen. Adjust the volume a little bit. There we go. Awesome. So if you guys haven't played Kahoot before, it is a trivia game. And what you'll do is you'll use your smartphone, your computer, and you'll go to www.kahoot.it. Once you've entered that address, you'll enter the game pin that's listed here. So 7987349. And once you do that, it'll ask you to enter your username. So you can make up one if you would like. And we have 6 questions about oysters for you guys to answer this evening. And I have this set up, actually, Kahoot's done something new where on your phone or wherever you're playing Kahoot, you'll see the question and the answer options on your screen. So you don't have to go back and forth between our screen and yours. You should just see it all happening on your screen. So hopefully that works for you all this evening. We do have a prize pack for you guys, some CBNERR hats and pens and tattoos, temporary tattoos for the winner this evening. So if you are the winner, we'll have you drop your email in the chat for us, or you can send it directly to Sarah and I, and we'll mail that out to you. But you'll get more points by answering the question correctly and then also by answering quickly. So the quicker you answer, the more points you get if you're right. So again, there'll be 6 questions that are about oysters. Some might have been covered in the presentation, some you might not know. So that's kind of part of the game here, but we'll wait a couple more minutes for some last minute people to go ahead and sign in if they want to play. And if you guys are a family, you guys can definitely play as a team. That's great as well. And if it doesn't show up on your phone as the question and answer choices, you'll have to read the question on my screen and then answer on your phone. Okay, guys. So we're going to go ahead and let me make sure everyone's checking the chat to make sure everyone's good to go. Okay, guys. We'll go ahead and get started. Question 1. How much oyster can an oyster filter in a day? Or sorry, how much water can an oyster filter in a day? Nice. Okay. Yep. So a single oyster can filter up to 50 gallons of water per day. So if you saw in that picture, one side of the water was really murky, didn't have a lot of oysters in it, and the other side was almost crystal clear and it was filled with oysters. And that's because they are those filter feeders and they create really great water and water quality. So nice job, guys. Check out our leaderboard so far. Next question. What are the odds of finding a pearl in an oyster? So this one's a little tricky. So we had a nice spread across the board, but it is 1 in 10,000. So it's not as rare as you think it might be. And oysters aren't the only animals that are capable of producing those pearls. A lot of different mussels can do it as well. And so that's something that people think every single oyster can produce one, but it's also not the rarest thing if an oyster produces one. So we'll see how we're doing so far with our scoreboard. Had some changes there, and we'll go ahead and move on to question 3. What is the name of the mussel oysters use to open and close their shell? Nice. So I briefly touched on it during our oyster cage. So that adductor muscle is inside of their shell to help keep it closed. And so whenever you're shucking a shell or shucking an oyster, you're trying to actually separate the that muscle from the shell in order to actually open it up, rather than going at it by the hinge of the oyster. So we have some people on streaks and some movement in the leaderboard. So question 4, about how many oysters are consumed yearly, and that's around the world? Yep. So 2 billion oysters are consumed yearly. There are people that aren't the biggest fans of oysters, and they can be prepared in many different ways, and they have different sizes. So there's many people out there that do enjoy consuming oysters, and that's a big fishery, especially here in the Chesapeake Bay. So the world does consume quite a bit of oyster. Nice. So it's so close race. We have a couple questions left. True or false? Oysters can change their sex. Well done, guys. So yep, it is true. They are capable of switching if they would need to be male or female based on their reproduction needs. There's quite a few animals that are able to do that. Awesome. Really tight race. Okay, guys, so we have one more question. How many oyster reefs, or how are oyster reefs and oyster populations being restored in the Chesapeake Bay? Nice. Everyone got that right. Nice job. Yep. So here in the Chesapeake Bay, we have different regulations put in place about when you can catch oysters, how many you can catch, what size they need to be. We're building oyster reefs and oyster habitats that allow these populations to grow and thrive. We even have oyster sanctuaries where harvesting is not allowed whatsoever and creating a truly protected area. So we're doing a lot for our oysters. We'll check out our winner. Nice job. In second place. Nice job. So thank you guys so much for playing, and whoever used the username Mountain Crab or that was assigned to you in the chat, or you can send it to Sarah and I your email address, and we can mail you that prize pack with some CBNERR stuff if you guys would like to claim that. That'd be great. And so I'm going to go ahead and stop sharing one more time for you all. And we hope that that game was fun for you guys. I know a lot of the younger members really enjoy Kahoot trivia, and we even have it for next month's April Discovery Lab. But that is what we have for you this evening. I hope you learned a lot about some of the diseases that these animals face, some of the different animals that live with our oyster populations, about the oyster itself. And if you have any questions, we would love to answer those. Sarah and I can stick around for a couple minutes. But we want to thank you again so much for attending, and keep an eye out on social media. We will be sending out information on our next Discovery Lab soon, so you guys can register for that. But again, thank you guys, and I hope you enjoyed it. I'm glad you guys enjoyed it. Thank you. Cool. So we got the email for our prize winner. Thank you. I'm glad you guys enjoyed it. Thank you guys.